Thank you very much, Andrew. I feel quite bigged up there. Thank you. And I think you may have covered some of my early slides, actually. So the point at which a cell meets a biomaterial, a material for use in medical devices in or on the body, is a very important moment that's represented here nicely in the scanning electron micrograph. And you can see the way the cell stretches out across the fibres and will then signal other cells, both within the tissue itself, perhaps within the organ, and also systemically, in order to mount a response to that interaction. And this can either go very well for the material, or it can mount a massive inflammatory response, which can then ultimately lead to rejection of the material. This is one of my favourite images across my 30 years because it nicely represents my career to date, which has been to look at ways to optimise this interaction, to better understand it, ultimately to improve tissue engineered product and medical device technologies as they interact with the tissues and organs of the body. So before we get on to some definitions, I'd like to start just first with a dedication to my parents, who are seen either side of myself and my siblings here, and also in their younger years. My dad has been known to comment on the fact that he doesn't really know where the science thing came from, to which my mum's response is, "Don't forget, I'm a retired palliative care nurse." And so today is really an answer to their question throughout the years of, so what is it you're working on now? And you can also see myself and my siblings in what we call the Morocco years. So we spent 3 years in my early childhood living in Morocco. My siblings recently reminded me that at about this time they actually dropped me on my head, lowering me down over a wall to retrieve something that they had lost. And so perhaps that explains also some of the science thing as well. Who knows? I'd also like to start with an early acknowledgement of the research students and staff who have worked with me across the years and have helped me in establishing a lab with an open science philosophy, which allows even very early researchers to come into the lab, secondary school students who are on placement, and then undergraduate students with placement opportunities, in order to experience the flavour of science, lab-based science, and infuse them and encourage them hopefully into future, that the wealth of possibility there is within science and beyond science as well. So you can see in the top right there, 2 secondary school students. One of them at the time wanted to be a lawyer, one wanted to be a chemist. And they did actually shift to study biomedical science, which I was quite pleased about in the end, although not through my coercion, of course. But here they are learning how to culture cells within our tissue culture facility, and learning that you can manipulate those cells in order to understand a little bit more about the human condition. So my students have got out and about as within their PhD and as postdocs. I think Grace drew the short straw, having to attend a conference in Hawaii. This one I think maybe from Bordeaux, but I've certainly been privileged to work with some technically skilled and highly motivated and hardworking individuals across the years who have made me look very good in all of the presentations I have had to give, including this one. And so let's move to some definitions. So I believe my dad's first request was that he understand the terms regenerative medicine and tissue engineering. So regenerative medicine is a multidisciplinary endeavour involving benchtop scientists through to medical clinicians and biotechnologists. And together we seek to understand better the reasons why disease and injury disrupt tissue and organ function, and also then come up with solutions to these failures through repair, replacement and regenerative medicine. So we're talking about things like coronary stents to unblock coronary artery occlusion. We're talking about intraocular lenses to treat cataracts. We're talking about the materials that are used within hemodialysis equipment for patients who have renal failure. And we're talking about other tougher nuts to crack as well, in terms of being able to ultimately take an individual cells, perhaps use some natural or synthetic materials, take some bioactive agents, and ultimately regenerate organs as they fail throughout the lifespan. So my research is within the tissue engineering area of regenerative medicine, which is represented here by what's known as the tissue engineering triad. So here we incorporate biomaterials and cells and these regulatory signals, and together we seek to use these in the repair, replacement, regeneration paradigm. Biomaterials themselves may be synthetic or natural. They may be made of harder materials, so these are metals. You might see those in bone replacement, in hip replacement and so on. Or you might require a softer material that's more flexible and malleable. And you can see there an example of a porous polymer. Or we might use ceramics, for example, in dental implants. And then the cells themselves may be autologous, so derived from the individual. They may be donor cells if autologous cells are not available. And we may also manipulate stem cells derived from the individual and encourage them to transfer into the type of cell that we actually want to replace the function of. And then regulatory signals may be required in order to perhaps perfuse from the device we're implanting for a benefit in treating disease. Or they may be used to control cell behaviour. And so together, or even in isolation, we may use these three aspects of this triad in order to tackle challenges of disease and injury. So we mentioned about the bioface in that first slide. I mentioned already that this is one of my favourite images. And you can see three things about this biointerface: the point at which the cell first encounters the biomaterial. So here you can see a fibrous biomaterial for use within an ocular implant. We've coated it with a bone mineral called nanohydroxyapatite, in an effort to encourage a cell nucleation site, so cells to migrate into the material and make it home. So that cell doesn't work in isolation, it works with other cells, as we've said, and it will start to give out secretory signals, which will then control that dynamic interaction. And there are three things that are important here. And it's what we will always do when we start to consider a new material. We will look firstly at what we call the biocompatibility. So does it do any harm? And that will be partly because of the material itself and its properties, and it will partly be due to the immune response that's mounted. The second is, does it do any good? So does it function in the repair capacity that we want it to? And then we also have to take into account the pathology that the device and the material itself is being used to treat, and issues of patient diversity. So the cell might arrive angry here, and it may already be part of a immune response. And so what we want is passivating materials that will help to moderate that response rather than making it worse. So we need to understand the pathology itself. We need to understand the biocompatibility and what we call efficacy or function of the material. Does it do harm? Does it do good? And we need to understand that we can first test in healthy, in a healthy cellular environment, but we also need to understand it in the site and in the disease condition that we're attempting to treat. So biomaterials began about 100 years ago as a kind of in a professional context. And we began with materials where the focus was really, could we get it to hide from the human body when we were using it in an implant as a bioinert material And we do sometimes see this as a property that we want in stealth materials that you might want to hide and then release so that it does good. For example, you might not want a material to be released in the stomach. You might want it to be released in the large intestine, and therefore you want a stealth approach where you hide it from the breakdown processes of the stomach. But on the whole, it became obvious that actually there will always be an immune response to a particular biomaterial. It will not, by its nature, really be truly bioinert, so truly hidden from the immune system. And therefore, we began to think about materials as biointegrative, so a term where we might say coat the material to improve the interaction. We might put in some drugs that we slow elute over time to also improve the function of the material. And then we move now really to perhaps a more hot topic, where at the moment we're beginning to design and see materials and devices come out that can read the tissue environment itself and then can respond appropriately. So not just incorporating a slow drug elution that will release as soon as it hits the tissue environment, but perhaps waiting, for example, with a pH trigger, so that when you get an infection, pH goes up and the appropriate antimicrobial drug is then slow eluted into the wound or the tissue environment. But that doesn't happen when it's not needed. It happens at the appropriate point. And so we've moved from bioinert through to biointeractive. And so this really kind of encapsulates where I've headed with my research, all about improving that cell biomaterial interaction, the biointerface, in order to replicate tissues and organs of the body. So both in a materials approach with artificial organs, but also in a biomaterials and bioartificial organ approach, where you have the cells and the materials and bioactives that you incorporate together. And what we're going to do for the remainder of the lecture is just look at a few examples throughout my career. Andrew has nicely summarised some of them quite well, actually. So we're going to look at a few of the ocular biomaterials and some of the visual repair approaches that I've been involved in. We're going to look at some of the blood detoxification and liver and kidney support systems. We're going to come back briefly to ocular biomaterials and then finish with a little bit of a discussion on these biointeractive approaches to chronic wound repair. So that's where we're headed. But first, I must talk about how I ended up at the University of Brighton. Andrew alluded to it somewhat. So I was finishing up an intercalated BSc at QML, and my supervisor at the time, Dr. Aronson, said, I know a Dr. Marcus Allen at the University of Brighton. We were interested in calcium channel activity. He does some patch clamping. Would you like to go and do your 11-week block project at the University of Brighton with Marcus Allen? So I was actually commuting at the time from Brighton to London, and so I thought that's a good idea. So I found myself at the University of Brighton learning how to patch clamp. And the reason I mention this, so patch clamping is a very interesting technique where you're able to lower a probe onto the surface of a cell, which you can see here. Thanks to Emma Ward for supplying the image. And you lower the probe onto the surface, and you can measure activity of channels within the cell membrane. And in this instance, we were interested in calcium channel activity. And so what happens, and you should be able to see that here. So you can see the calcium channels switch on, calcium floods into the cell, and you see a change in signal. And you can see that we've got a fluorochrome where you get a fluorescent green signal when calcium channels open, and calcium binds to the fluorochrome, and you see this green fluorescent signal. So that nicely demonstrates calcium channel activity. Why is this important? Well, we can look at upregulation of calcium channel activity as a marker for things like neurotransmitter release or modulation of pain pathways, which becomes important when you're looking at the development of materials and devices to get in the way of upregulation of pain pathways or to be involved in nerve replacement type activities. But that's not really why I'm showing you this slide. I am now in the office that is adjacent to the office of Marcus Allen, who retired recently. And as he did so, he walked in and handed me back my research project, complete with the sticky note on it from 30 years ago. So, and I was able to insert it nicely here. And it was at this point when I was considering what else to do. I'd already decided not to go on to the clinical portion of the degree. And I sat down in an office with, who was then Dr. Andrew Lloyd, and he said, would you be interested in coming and doing a PhD at the University of Brighton? And to which I thought about it and I said yes. And so I found myself at the University of Brighton, never expecting to still be here 30 years later. But here I am. As I told the students at the beginning of the year, something always came up. And so here I am. So I started my PhD, and this is quite a wordy title. So I asked ChatGPT to come up with another one. And it said, you need something. It said, first it said, congratulations. I said, to come up with another one for an inaugural lecture, to which it said, oh, congratulations. And then it said, it needs to be dignified but accessible. And so I thought that was a great comment, but it didn't come up with anything useful that to me seemed dignified, useful, or actually correct. And so I came up with this, which was improving the biointerface, which we know what the biointerface is now, improving the biointerface for better retention of a tissue-engineered cornea. So that's what I was interested in. If you look into the eyes of the person next to you, you won't be able to see the cornea, because it should be transparent. So you can see quite nicely here an eye, you can see the white sclera, you can see the coloured iris and the pupil, the gap in the middle. The cornea arcs over the iris and is continuous anteriorly with the white sclera. And you can see my nice brick-like approach to drawing, so that's straight from my thesis, drawing the structure, the 5-layered structure of the cornea. There's an outer epithelium which is coated by a tear film, continuously washing the cornea with the blink reflex. You've then got a stromal layer in which you have cells represented by these black dots here that are fibroblasts. They're corneal fibroblasts called keratocytes. I spent a lot of time with these cells. They are my favourites in my PhD. And here you can see a second one of my favourite images, which is fluorescent staining of an adult keratocyte isolated from a time-expired donor cornea. And the reason why it's one of my favourites, even though it's slightly blurry, is that I actually had to take the roll and take it down to get it developed and tell them to also develop the one that was completely black because that was my negative control. And so these cells reside in the stroma and they are responsible for maintaining that a really tightly structured collagen fibrillar matrix. We've talked about the fact that this must be transparent, and therefore those are effectively housekeeping cells. They actually sit quiescent and can sit quiescent and not dividing for up to 2 years. However, they kick into motion when there is any insult or any repair that needs to be undertaken. So that stromal layer is important. It's separated then from an inner endothelial layer by Decimus membrane. And so I spent a long time looking at the wound repair response and then looking at different biomaterial strategies that we could use in order to optimize that wound repair response. And for that, I was awarded a PhD. We'll unpack a little bit what I did. So here you can see a cross-section through an eye. You can see at the front. Let's see if I can do this. It's going to work. So you can see at the front there, this transparent cornea, continuous with the white sclera round to the back. So the cornea is responsible for the majority of refraction of light, so bending of light as photons of light pass through the eye. The lens then here forms the rest of that bending of light so that you get light focused on the retina at the back of the eye. So you can see, hopefully, so these are avascular structures. It's an immune privileged site, and you can see, hopefully, why that line that line should remain transparent so that light can pass through. And it's a problem if it doesn't. So corneal opacity is responsible for, is one of the big five, it's the fifth most common cause of blindness globally. And normally you can successfully have a donor cornea. However, for some patients, for those with chemical burns or other autoimmune diseases, they will not tolerate a donor cornea. And so we need alternatives. And so one of those alternatives is a polymer plastic based system. And you can see an example there. So this is like a mushroom-shaped artificial cornea, also called a keratoprosthesis. And you can see there's a central clear optic made of, and then there's a skirt-type material which anchors this device within the eye. And so what I was interested in was the skirt material. We need an integration of these cells and a material which enables that, but then we also need a material in the middle that retains clear. And what I found in culturing the cells was that they actually lose divisional potential over time in culture. So as they age in culture, they actually exit the cell cycle. Here you can see, so Chi67 is a stain. It basically measures divisional cells. So you see a decline in divisional cell potential as the cells age in culture. And this is known as cellular senescence. So at each point, the cells are permanently exiting the cell cycle until you get to the very bottom there. Now, this wouldn't necessarily be a bad thing, and it can have some senescence can have some protective benefits. But in this case, senescence also alters the performance of the cells. So the cells switch into a more inflammatory state, and it can then produce a decline in the repair response, which isn't great when we're looking for a material that will work here. And so what we looked at also were different ranges of porous polymer skirt materials, which could then be modified in order to promote this wound repair response. So we're able to publish 3 papers out of that work, and I then went on to a postdoctoral position with Andrew Lloyd and Brian Ty at Aston University, where we were continuing to do this work and looking at organ culture models and the development of organ culture models in order to test materials in a more physiologically appropriate environment. And I was able to watch ophthalmologist Chris Liu come in from a day of surgery on eyes at the Sussex Eye Hospital and spend about 10 minutes really niftily inserting our implants into these organ culture corneas that we were using. So shortly after that, these two came along, so Jack and Anna, and I became a full-time mum and a part-time scientist for a bit. And they were, although it may have been more kind of nearly full-time and full-time. So, but these guys were definitely the priority. There were a few things that were really important about that time, some of them not under my control. One of them was a very successful job share with Dr. Carol Howell, in which we were one researcher between us. The other was I was fortunate to work in a very active research group, firstly led by Andrew Lloyd and then led by Sergei Mikolovsky, in which they supported our part-time job share, and also it was a very active environment where we had researchers. It was an international stage for research. We had visiting researchers coming in. We were encouraged always to get out and about, presenting our work, and also given the possibility to write grants and develop our credentials as researchers in our own right, building our own networks. And so out of that time, I was able to continue to work on some eye-based research. And the first I want to show you is this work done with Elsie Folk. And it was again looking at the cornea, but at the epithelial layer, so that outer layer. And in order to look at Elsie's work, we'll first look at some of the foundational issues in inflammation at the epithelial stromal interface. You remember we talked about those fibroblasts in the stroma, the keratocytes. So now we're talking about the epithelial cells. So unlike the keratocytes, these are continually dividing over time. And the epithelial cells, when there is an insult or an injury, they will mount what's called an inflammatory response, and they will produce inflammatory cytokines. So here we have interleukin-1 alpha and beta. These epithelial cells will start signaling other cells to induce an inflammatory response. And they will also talk to the keratocytes, which have receptors for IL-1 beta. The keratocytes are activated and they in turn start producing inflammatory cytokines, which then upregulate the cells in terms of an inflammatory response so that they can then start to heal the wound. Now that will then be moderated by an anti-inflammatory response. And if that doesn't happen, we can get a hyperinflamed environment and we can shift into a chronic non-healing wound scenario, which is bad when it comes to the use of materials-based approaches. So what Elsie was interested in was the challenge of corneal limbal epithelial stem cell deficiency. And this is, so you can find the stem cells, the corneal limbal epithelial stem cells, in the limbal region of the cornea. And you can see here, this eye here, I'm not sure if you can see that, but the red dotted line shows you where the limbal region is. And what that limbal region does, those stem cells, well, firstly, they produce progenitor cells, which then allow the continual production of epithelial cells over time to replace the corneal epithelium. But they also hold back the stroma. So should those limbal stem cells fail, the stroma will, the stromal structure will walk across the anterior, and you'll get fibrous scar formation and obviously lose vision. Now this can be treated with a graft. However, there are challenges. There were challenges at the time with tissue availability and rejection of the transplant, and also this inflammatory response which contributed to that. So what Elsie was looking at was developing a short peptide, which it's quite a wordy title here, but I'll unpack it for you. So this was an IL-1 beta receptor antagonist, and what that did was it bound to the receptors that normally took the IL-1 beta and promoted that inflammatory response, both in the epithelial cells and those fibroblasts. So it muted the response. She then coated that on a contact lens as a carrier type matrix that the limbal epithelial stem cell graft was then grafted onto the eye, or that would be the potential final result. And here's a little bit of data for you. So hopefully you can see this. You can see that when the peptide is present, which is this bit here, this is the inflammatory response, so this is one of the cytokines. When the peptide is present, we get a muted inflammatory response. When the peptide isn't present, we get an augmented inflammatory response. She also did some work showing that the stem cell fraction was maintained over time. So this was a really nice result. Elsie published her work. So bear in mind the inflammatory cytokines here. So this is contained within that corneal matrix. But as we move forward, I began some work within my postdoctoral position with Carol, looking at adsorbent materials which could absorb these inflammatory cytokines, particularly with regard to more systemic issues such as life-threatening sepsis, where extracorporeal systems have been developed in order to use adsorbents that could grab out these inflammatory cytokines. And so we worked with a company who developed these adsorbent materials, and they were unique because they had a porosity which wasn't found in other adsorbent materials at the time, which was in this nano range, and we had already demonstrated that inflammatory cytokines could be removed, bacterial toxins could be removed. And also we could tackle some tricky kidney and liver toxins, which normally travel in the blood, bound to the dominant plasma protein albumin. And what we also did was look at, can these beads be made in an extruded form so that you don't get the pressure drop with beaded systems? So that was the backstory. We started to look at the challenge of blood detoxification. And then I went on, I think I tossed a coin with Carol actually for the writing of this grant. I went on to write a grant where we we took what we knew about the adsorbents and looked at the monoliths as a potential way to augment kidney dialysis. So kidney hemodialysis is a life-saving therapy for patients whose kidneys fail. And what happens is the blood is withdrawn, it then passes through a dialyzer. You can see the cartridge there. Blood flows through the fibres of the dialyzer, and in countercurrent, dialysate is pumped. And what happens is excess fluid and some of the toxins are removed and the blood is returned to the patient. That keeps the patients alive over time and is a successful therapy. However, there are a few issues. And one of those is that some of the larger uremic toxins, so toxins to the kidney, they fail to be removed by this type of system. And we did some work with Andrew Davenport and a student, Sankina Kandusi, this was with Yishan, and we did some work looking at these two particular albumin toxins called indoxyl sulfate and p-cressyl sulfate. And here you can see the work was looking at vegetarian versus non-vegetarians. These are molecules that are derived from gut bacterial breakdown of particular amino acids, tryptophan and tyrosine. And so we were interested in looking at these molecules, but what we found there was that these molecules are not removed during hemodialysis. And so this was my first grant, which was funded by the NIHR, and we were looking at whether we could replace dialysis, but whether we could place an adsorbent cartridge in line with dialysis in order to augment its removal capacity without disrupting any other blood biochemistry markers negatively. I've included a little bit of data just so you can see what we saw. So in the first grant, which is called DART, we saw for all of the markers we looked at, so here's indoxyl sulfate. Here you can see patient blood pre-dialysis, patient blood post-dialysis, and then we perfused it through these small prototype monoliths over time, and you can see hopefully that without our adsorbent there is no removal. There's certainly no removal with dialysis, but then as we flow the blood through our system, you can see a reduction over time. So this was significant, and I went back to the NIHR for more money to try and scale up the system, which was successful. And the reason it was successful, well, one of the reasons was I went to an event where the funder was present, and I was talking to him about the grant, and I was saying I'm going to ask for a little bit more money. The first grant was just short of £100,000, so I thought, well, I'll ask for a bit more money. And he said, oh no, you want to ask for much more money than that. And so at that point I began to think big, and at that point I was still in the position where I wasn't sure if the first grant had been a fluke. And so it was it was a story about making sure you talk to funders and making sure you turn out to events in order to talk about the development of projects. It can be the difference between success and failure. And so towards the end of that second grant, I was then in charge of an FP7 IAP, Industry, Academia, Partnership and Pathways project called Acrobat. And this was a broader grant with 3 academic partners in the UK and 3 companies, 2 in Germany. And I really saw the benefit of the interaction of industrial medical device companies with effectively our benchtop science, and we were also working with clinicians at the hospital. And here you can see a visitor from Albertech in Rostock in Germany. They're a liver dialysis company, and this was a visitor in our lab, and he was showing us how to use a hemodialysis machine in order to test our prototypes, test pressure drop, and test with more clinically relevant parameters. And so we were able to test the bead system and the monoliths in this scaled up system, and this allowed us to move from the data we'd seen in the small systems through to these larger systems. So here you can see this porosity, here you can see the decline in these toxins over time, and we found that as we scaled up we were able to replicate that at clinically relevant pressure, flow and timings. And so out of that project, we had also I'd also been running in parallel a project with Rajiv Jalan's group up at UCL, looking not at blood perfusion devices, but looking at the use of these adsorbents as oral adsorbents for treatment of liver cirrhosis. And we had a little bit of Brighton Business Investment funding to do some early studies with Rajiv's group. Tiny Beads of Hope is the newspaper headline from UCL's press release, not my words. And so what this project began to consider was the gut lining and the fact that as cirrhosis of the liver progresses, you get something that's called gut dysbiosis, which has an impact on the liver, because the vasculature from the gut flows straight to the liver. And so you get a reduction in gut barrier integrity, and here you can see that you have a change in tight junctions, so that bacteria that should remain in the gut then have the potential to actually migrate across into the surrounding tissue. Those bacteria tend to be a more pathogenic variety, and you get release of endotoxins, so bacterial toxins, and upregulation of inflammation. And this causes problems for patients who are already perhaps immune compromised. And what we found in our early studies was that we were able to reduce the symptoms that occurred around this process. And that went on to a Horizon 2020 grant called CarbAlive for clinical studies in a multi-centre European-wide clinical study looking at safety and efficacy in patients, and led by Rajiv's group at UCL. So within the Acrobat project I previously mentioned, we were also looking at porous polymer materials, and this was based on some of Yishan Zheng's PhD and her early work, where she was able to go on to comment to Lund University in Krems on a project led by Sergey, and she was able to learn a technique called cryogylation, in which you can make these porous polymers that have connected macroporous structures that allow fluid to flow, and that structure is made by the use of ice crystal porosogens. So within the Acrobat project, we built on that with Ganesh in Gavle's work, and here you can see the porous domains here. And what Ganesh was doing was working with Les Bailey at Cardiff, and also with UCL, where they were looking at whether we could not just incorporate adsorbents within this structure, but also bind antibodies against bacterial toxins. And I won't go into the detail of that. But out of that began this idea that perhaps we could use these materials also in other clinical applications, particularly with another cirrhosis-related aspect, which is how can we replace the failing livers? So we talk about hemodialysis as a life-saving therapy for kidney failure. There is no equivalent for patients with liver failure. They rely on transplant. Otherwise, there are issues. So there are some liver dialysis treatments, but they have been shown in trial not to clinically extend patient lifespan. And the challenge is this: you must have a detoxification function, but you also must perform the synthetic and metabolic functions of the liver, which are quite complex. And a number of bioartificial liver clinical devices have been tested through many millions of pounds and right through to clinical trial, and have not again shown any benefit to patients in terms of extending lifespan. So what's a bioartificial liver, you say? Well, so it's not just a dialyzer, but it incorporates a cell component here. And so we began to think about whether these macroporous polymers could be used. And so here you can see some of Flavia Bonalumi's work, which was supervised by myself and Irina Savina of Cryogel expertise, and also engineer Cyril Kruer. And what Flavia did was she stacked these porous polymer discs, incorporated cell components, and showed that she you can see those in green here in each of the stacks, and she showed that she could maintain these over time, and she surface functionalized those cryogels in order to benefit cell interactions and then reduce any biofouling potential. And here you can see her work. She compared a solid monolith structure versus this stacked system and did some work with Cyril to model the velocity profile of blood cells flowing through the system. And you can see a little bit of the functional performance of these devices. So we want to be able to synthesize things, and you can see that albumin is made by this system. So that's the column versus the stack. We also want to be able to process some of the toxins, such as ammonia. And you can see that ammonia is processed over time, and you see the production of non-toxic urea from ammonia here, and that's rising up. So we had success here, but we were also aware that there were other factors that affect the success of bowel performance. And we were testing in a healthy setup. But what happens when the metabolic products from a patient whose liver is failing build up? What does that do to the success or failure of these kind of devices? And so I met Richard Farragher in a corridor, as you do, and started chatting about the potential that senescence could play a role in the failure of these bowel devices, and whether that could be a potential hypothesis for Neda's PhD. And so what Neda did in her PhD was begin to look at whether it could be senescence of these cells on exposure to these toxins that caused these devices to fail. And here you can see some of the data. So these are cells alone, and you can see this is just a marker for divisional potential of the cells. You can see when they're exposed to toxins, so they're alive, that's those blue dots, but you can also see that they've exited the cell cycle. And then we were also able to work with some senescence protectors called, these were analogues of resveratrol, which were able to protect the cells from senescence. And you can see that there's a return of the green fluorescence. So what Neda found was that exposure to these liver toxin cocktails does cause senescence. And they can be protected against. So you can see a little bit of that data here. For some of you, you can see this is a measure of cell senescence. So that's a bad thing. You can see exposure to liver toxins, it shoots up in red there. And then you can see that the resveralogs protect against. And then we also wanted to look at some synthetic function. And you can see here, again, liver toxins reduce synthetic function over time, but that's regained when we use this protective resveralog. And so that was really pleasing results that came out, and then supported another MRC grant that was led by Richard. So that takes us on to another very productive grant, which was a small grant, but it was funded by the British Council in association with the Department for Business, Innovation and Skills. And in this project, we worked with Drexel University and Northwestern Polytechnic University. And one of the highlights of that was that it allowed early career researchers from master's level, PhD, and postdoc to go to these different centres and exchange information about biological testing of adsorbent materials for the removal of contaminants that could cause harm. It also meant that we could head to these different centres. We had 3 different symposiums, and here you can see our project officer, Gavin Gunter, who's working hard in Xi'an, testing the food for us. He did bring us in budget and on time, so he did an excellent job. And you can see Yishan and myself, who was the best tour guide ever to go to Xi'an with. We got the highlight of seeing the terracotta soldiers. But one of the most important things I think that came out of that project was that when we went to Drexel, I sat in on a presentation, and they showed a slide of a polymer with a Maxine coating. And it was transparent. And because of my experience in eye, I was always looking for things that were transparent that could be used as artificial corneas or perhaps within the lens system. And so what are these maxines? Well, they're related to the graphenes. So they are two-dimensional materials. It's a family of materials, and they are represented by this general formula. So M is a transition metal, X is a carbon or a nitrogen, and then TX is a surface functional group. And we worked with one called Ti3C2. But there were key properties. One was the transparency that I'd spotted, but also that these materials were conductive, like graphenes, but they were hydrophilic. And also they were flexible, and you could moderate the surface chemistry. And that was useful, particularly with regard to ophthalmics. You can see here some SEMs of the stacked layers of these nanomaterials, and that they can be used in different scenarios. So you can spray coat things, you can spin coat, you can print, and so on. And so they were highly malleable and flexible materials. They were conductive, they were transparent. We began a study with colleagues at Drexel looking at a particular tough nut to crack, which is the wearable artificial kidney, and what to do with urea. And I won't go into the details of that for time, but we demonstrated that you could use these as multi-stack systems, and you can incorporate and intercalate urea into these stacks. And actually, it could potentially be used in wearable artificial kidney technology, where you have dialysate that must be refreshed. And one of the tricky things is what to do with the urea. So that was really nice, and it was licensed to a kidney dialysis company. But I was interested in eyes, and so the nice thing about this was that we had a good group of international supervisors, one in material science experts, so Yuri Gagotsi at Drexel University. We're also working with Rayner Intraocular Lenses, it's a local company, and then myself. And so what we did was then write for an MRCI case to look at the development of materials for intraocular lenses. Now, again, this is a very successful implant technology. So should you get a cataract, you may well have had a successful implant here. So what happens is here's the lens, here's the cornea. The cataract develops within the lens, and you disrupt the visual acuity. And the surgery is to implant this intraocular lens here. So you remove the cataract and you implant a foldable IOL into the lens, and you get a return of visual acuity. One of the problems is that you lose adjustable vision, so your ability to focus on near and far away objects, so you rely on glasses. The second problem is that residual lens epithelial cells that remain in the capsule sometimes track across the back of the eye, and you form a secondary cataract over months to years. And so what we looked at with these Maxine materials was to tackle the first issue, which was whether we could incorporate an electrically conductive material which on signaling could then be used to change the power of the lens and allow focus at the back of the retina. And this is Emma Ward's PhD, which was funded by MRC. And she did some really nice work optimizing a spin coating technology with Maxine, showing that it could both remain transparent enough to be used to coat an IOL, but also would be electrically conductive and could be potentially used in this setup. And here you can see nice proof of concept work here. So it was a sandwich approach where you had a Maxine coating, you had liquid crystal, and then you had another Maxine coating. When she applied a charge, hopefully you can see how a blurry image then became clear. So that was Emma's PhD. We also wanted to look at that second aspect, which was secondary cataract, and which is also called PCO, or posterior capsular opacification. And here you can see what I've just described. So this is a capsular bag. You can see the lens. Any residual epithelial cells may actually induce a fibrotic response. So they think they're repairing a wound, but they induce a fibrotic response and they grow a membrane across the IOL, and that's not good in terms of visual acuity. And so what Grace's work did was look at whether Maxines could be used as coatings which were fibrosis repressive. We also wondered about whether we could use drug elution strategies as well. So the idea was that you coat the lens and it represses inflammation. And I've got a little bit of data just to show you. Hopefully you can see. So this is instigation of a cell model of fibrosis. So we stimulate the cells to become fibrotic. Here you can see no green means no fibrosis. Our material, no fibrosis. Fibrosis here, when we used our material and induced a fibrotic response, you can see that there's a repression of this fibrotic instigation. And so that was some nice data. There's a lot more data there, produced by Grace and published within her PhD. And so Emma and I've thrown these images in thanks to Emma and Grace. So we talked about regenerative medicine being a multidisciplinary endeavour. And here you can see science meeting art. This is an international competition, and both Emma and Grace were prize winners. So this is run by one of the alumni out of Drexel, Babak Kansuri. So I really like Emma's visualization. So that's her liquid crystal Maxine layer under light microscopy. And she's used it to visualize. It's a bit space-like, where we have science astronauts perhaps visualizing on a spacewalk the wonderful world of Maxine. And then Grace produced an image which for all those climbers out there was to do with the kind of snowy caps and the fact that Maxine scientists are beginning to climb the snowy peaks of discovery within the Maxine world. So really nice demonstration of the creative nature of Grace and Emma and of the science community as it looks to innovation. So we finished now on the last project, which is to do with the challenge of chronic wound repair. So the normal wound repair response, which we've been talking about a lot, moves from a blood clot, hemostasis, through to an inflammatory response, and then through to remodeling and proliferation to remodel that early matrix that's laid down, and then you get wound closure. However, for many patients, particularly those with comorbidities, so other diseases like diabetes, you have problems with the normal wound repair response, and they end up with chronic wounds. What happens there is that you get stuck in this inflammatory response. And so this is the work of Sertan Kizelos, who's just finishing up his PhD. And hopefully you can see a normal repair response with this. Let's go back. With this scratch and then closure of the wound. And then if we start this one, so this is a stressed condition, much like you might get in a diabetic wound. Hopefully you can see that the stress condition is much slower in migratory capacity. So Sertan's done a really nice job of using that modelling in order to look at the development of stimuli responsive chronic wound treatment device that could potentially both read the wound and then respond appropriately. And here you can see a nice graphical abstract. So here you've got the chronic wound, which is stuck in inflammation, and you've got all sorts of inflammatory signaling going on here. So what Sertan did was develop it's an osmolyte eluting microgel technique where this positive influencer of inflammatory stress is contained within the matrix. You also have a Maxine coating, which he showed repressed the inflammatory potential of the wound. And then you get slow elution of this osmolyte from this microgel over time. And what Sertan found was that this device then showed improved wound closure rates, reduced inflammatory markers, and reduced apoptosis. And he's now just finishing up some work looking at whether we can use some of the inflammatory upregulated markers in the wound in order to then trigger the elution of these microgels over time. So I mentioned things like pH. You can also use protease activity by coating some of those microgels and then having those proteases break down the proteins that encapsulate and so on. So that shows you how we've gone from bioinert materials through to more interactive materials that can read and respond in real time. And that gives us a shot at much better treatments for these types of fibrotic and chronic non-healing conditions, which hopefully you've seen scattered throughout the different pieces of work that I've shown. So we reached the end. I'd like to acknowledge, I've already acknowledged obviously the research students and staff within my team, and here you can see some of the colleagues I've worked with across the years on the projects that I've mentioned, but there's also a number of other colleagues who aren't mentioned here who I've worked with both in my doctoral college role and across the university, who've really helped me to understand the breadth of research and what it means to make a contribution, to think about gaps in knowledge and so on, not just within my discipline but in other disciplines which both touch on my own and in some ways are completely removed from my own. And then I'd like to give a final thank you to these guys. So Faith and Family have been a really important part of my journey as a scientist. So I'd like to acknowledge my best friend, Michael Sandeman, who's also my husband of 31 years, and our tribe here, so Jack and Anna and their beloveds. And with that, I shall hand back to Martin. Thank you very much.